So welcome to part two of this Working with eBird Status and Trends Data Products webinar through Birds of the World. This one will be focused on applications of the status data products. I'm Matt Strimus-Mackey. I'm a data scientist on the Status and Trends team. And among other things, I manage the R package that you use to access Status and Trends data products. And then we also have joining from the lab, Tom and Andrew, and I'll let them just introduce themselves.
Hi, Tom Auer, lead geospatial data scientist in the Center for Asian and Population Studies and help lead the production of the Status and Trends products.
My name is Andrew Stillman. I'm a quantitative applied ecologist working in the conservation science group here at the Cornell Lab. And my job is to take eBird data products and make them useful for conservation, working with a number of different partners and conservation agencies. Back to you, Matt.
Thanks. Tom and Andrew are going to be helping with questions as we go through this. And I just want to remind everyone, because the group is so large, we're not going to do, people putting up their hands and us turning on the video for those people. Instead, just please use the Q&A button to ask questions, and don't use the chat for questions. Use the Q&A button for questions, but you can, post other things in the chat. All right, so let's get right into it. All right, so this is a bit of an overview of the plan for today. I want to start with a recap of what we covered in part one. It was a little rushed, especially towards the end, so I just want to go over some of the things to make sure we're all on the same page. Then we'll address some of the questions that came up in that Google form from part one. And finally, we'll get into the bulk of the material, which will be covering a couple of applications in R, where I'll do some live coding. And then with whatever time we have left, we'll answer more questions. So it should go for 90 minutes officially, but I think all of us can stay for an additional 30 minutes until 2 p.m. Eastern if there's a lot of questions. And then I just want to remind you that this is part of this 3-part series. So last week at this time, we had an introduction. That's available on YouTube if you weren't able to attend that one. And then this week, we'll talk about status applications, or the status applications, so the relative abundance data products. And then next week at the same time, we'll talk about trends applications. Okay. So I want to start by just as I was saying, recap some of the content from part one. So I think that the first thing is I want to acknowledge that there were a number of people who pointed out in the Google form that I could have done a better job providing motivation for the applications and examples. So, why we're doing what we're doing. And also I could have done a better job helping you as attendees build a better intuition for the data products. So that was super useful feedback for us to hear. And with that in mind, I do want to cover, just talk about relative abundance again, just to make sure we're all on the same page about what that is. So ideally, what we'd like to know is the actual density of birds in an area, or the number. So for example, like there are 4 golden eagles in this 3x3 grid cell. But eBirders are just reporting how many individuals they detected, not how many are actually present. So some eBird checklists might report 4 golden eagles for that area. Some might report 1, and some might report 0. But since there's always that component of detectability, and it's not, most people don't have perfect detectability, we can't estimate the true abundance. Now, we try to minimize the impact of detectability by predicting to an observation by an expert eBirder at the optimal time of day and for the optimal weather conditions for every species. But that minimizes that effect, but it doesn't totally remove it. And this is why we refer to our estimates as relative abundance. They're proportional to the absolute abundance, but that there's some proportionality factor which is determined by that detection process. So this then leads to a related metric that we introduced in part one, called the proportion of population. So we saw in one of the examples last time that if we sum up the relative abundance for all the cells in a given region, so for example, a state, and then divide that by the sum of the relative abundance across the whole range of the species, you have a ratio of two numbers, and that detectability proportionality factor sort of cancels out, and then we're left with an estimate of the true proportion of population within the region. So although we can't get at true abundance, we can estimate the true proportion of the population. And then we'll see today that proportion of population metric is totally critical if you're trying to compare status and trends data products between species. So the reason for this is that detectability can vary widely between species and also between times of the year for the same species. So some species are extremely easy to detect. You're almost always going to see them or hear them if you go out and they're in a certain habitat. But then other species might be like super shy or quiet or extremely hard to identify. So those will have much lower detectability. And then because of this variation in detectability, we can't really compare relative abundance between species because it's affected by the detectability. But we can compare proportion of population because it's a ratio. Okay, so that's sort of all I wanted to say about relative abundance and proportion of population. I'm going to let Andrew talk a little bit about the regional summary information now.
Talk some more. Thank you, Matt. So as Matt mentioned, these proportion of population metrics carry a lot of information. And there are proportion of population rasters that are available from the eBird STR package. And so if you're interested in kind of calculating regional summaries of proportion population, you can do that by simply summing the pixels within some sort of polygon using the R package. But there's also regional summaries of proportion population that are available in other ways, including on the website. And so I wanted to share my screen real fast. And we're going to head over to the eBird Status and Trends website. Matt, can you see it right now? Good, perfect. Yeah, so let's just go over to the eBird Status and Trends website, and I'll put in an example species like blue-winged warbler. Let's see if that pops up. There it is. So what you're looking at is the relative abundance map for blue-winged warbler. And there's a couple of different ways to get these regional percent of population stats. One way that we talked about during our last meeting was if you go over here on the right-hand side of your screen and scroll down, there's this draw shape tool. I can draw a shape on this map. And the summaries that pop up are percent of population estimates within the region that I just drew. And these are really useful because percent of population or proportion of population for land managers or for people tasked with working in the conservation biology realm, they give you a sense of the stewardship responsibility that you have for a species in your region. Now, at the species level, you can also download regional summaries of these percent of population metrics for every country, state, and province that the species occurs in. And you can do this by going over here to the top right of your screen on the Status and Trends website, clicking Downloads. And there we go. So you can see it starts over here with abundance downloads. We talked about these last time. There's range map downloads. Toward the very bottom, there's trends downloads. But this section right here, regional range and abundance downloads, this downloads a spreadsheet giving summarized percent of population or proportion of population metrics for every season in every state, province, and country where the species occurs. And so if I click to download this, it just downloads a nice CSV, which is it's a file that can open in something like Microsoft Excel or a text format file. And it will give you columns with regional summaries for that species in each of those regions. Now, if you live in the US and you're working with US states, There's also another way to access similar information. In this case, over here on the eBird Status and Trends website, it's available at the species level. But we've also taken these same estimates and summaries and collated them at the state level. And so using this website right here, which I'll put a link into the chat later, you can download a spreadsheet with a row for every species that occurs in a state, and then seasonal percent of population estimates for that species, along with some other metrics. Like one that seems to be useful is the maximum percent of population that each state achieves over the course of the full annual cycle of the bird and the week in which that maximum value occurs. Scrolling down on this website, there's little videos and tutorials about how to use the data sets. And then down here at the bottom, you can click to download packaged summaries for your state.
Can we just do a quick live answer for a couple of questions?
Sounds good. Yeah, I'll stop sharing. We'll answer a few questions.
So we had a couple of questions about the biases related to data density in the proportion of population specifically, and then also about the relative abundance. And so yes, the quality may vary with the data density, but the proportion of population should help adjust that somewhat. Although if the underlying relative abundance estimates aren't great, then the proportion of population isn't going to be great. But when we're controlling for observer expertise or things like that, that is controlling across space as well. So hopefully that's not too much of a bias issue. If you get into, excuse me, areas with very low data density, you know, it's kind of, it's got to pass the smell test and buyer beware, like, you know, regions that are obviously undersampled by eBird, like Russia or Central Africa or parts of the high Arctic, you know, you're kind of on your own there in terms of the quality. We try to control extrapolation as much as possible, but it's not going to be perfect.
Thank you, Tom. Back to you, Matt.
Cool. Okay, so next I wanted to talk about, or to just address a few of the questions that came up through the Google form answers. So I'll just kind of like rapid fire go through these. Someone asked, are there other data sources used beyond eBird for these status and trends models? For example, the breeding bird survey in the United States. The short answer to that is no, but a lot of those other data sets end up in eBird anyway. So for example, something like 3 to 5% of BBS routes, breeding bird survey routes, are in eBird. So if they're in eBird, we use them, but if they're not in eBird, we're not sort of adding supplemental data sets. There was a question about how do you download multiple species at the same time? That eBird ST download status function doesn't allow you to download multiple species at the same time, but you can just write a loop or something and loop through the species and download them. And we're going to do that in the R section shortly. There was a question about how do we avoid bias towards rare birds that people target? So, you know, in the most extreme example, you can think of like a mega rarity that shows up in some place, and then like hundreds of birders go and see it. So you just end up with like a massive number of checklists with this very rare species on them. So first of all, we only use complete checklists. So people are indicating that they're reporting every bird they were able to detect. So for example, they're maybe reporting that super rare species, but they're also reporting all the common species. And then also we do apply some spatial and temporal subsampling to the data, which essentially like thins out the data. So, you know, you might have 200 checklists for that one rare species on like one day in one location, but we, through the subsampling process, will remove almost all of those and just have like one checklist for that spot. Okay, there was also a bit of confusion about the release cycle for these data products and how that relates to reproducible research. So traditionally, we've released updated status data products annually, and the next release will be in early 2025, and those will be predictions for the year 2023. Then after that, the next release will be in fall 2026, and then we'll likely go to a every other year release cycle rather than the every single year release cycle that we have done. On the trend side, we're not going to be releasing trends this year or next year, but we do intend to restart releases on some regular schedule, which is sort of to be determined at this point. At the moment, we're doing research to improve our methods for trends. Now, the R package only works with a single version of the data. So the current version of the R package only allows access to the 2022 data products. And then when we release the next, like in early next year, when we release the new data products for 2023, we'll release a new R package to work with those data products. We don't post data products from all previous years indefinitely. So if you need to retain access to data products from a previous year, for example, you have a paper or something like that that you want to be able to reproduce, we just suggest downloading the necessary data products and then just keeping them on your local machine as like an archive. And I think there was a question about this in the Q&A too. If you need to, you can always install a previous version of eBird ST if you want to access, or if you want to work with an older version of the data products. Okay, I think I'm going to let Tom answer a couple of more questions, so I'll pass it over to him.
Thanks, Matt. Yeah, there were a few questions that were easy for me to answer, relatively easy. I'm going to start with what are the minimum data requirements for running a species? For the status data products, there's kind of this bare minimum floor of 200 complete checklists and having a spatial density of greater than one checklist per 100 square kilometers. I realize that this is hard for people to then go to the eBird website and have any sense of whether or not a species is modelable. You can probably bump that up to about 500 checklists. And that might be, kind of where we're starting to talk about it possibly happening. But if that maybe applies more to a smaller range species, obviously, than a globally ranged species. That bare minimum doesn't mean that it's going to perform well enough to pass review or cover enough area. So it's not a guarantee, but it's a starting point. The volumes for trends analysis are about 4 to 8 times that much data. So it just requires a lot more data to understand the trend over time. So we need more data for the trend analysis. There were a couple questions from folks last time about high seas species. Let me share my screen here real quick. And okay, so I just wanted to put up an example of a species we have modeled. This was a test run, it's not the final version, but over open water and land, which is one benefit. Red phalarope, uses a fair amount of land as well as water. But the point of this animation is to kind of show two things. One is the kind of spatial extent of where we feel comfortable making predictions based on the over open ocean data that we have available right now. So obviously the species winters over a much broader chunk of the ocean, but we are picking up places where it is wintering closer to land. And then you can also see that there's probably some extrapolation in the Arctic Ocean during the summer months, as that's an area with low data density as well. But this year we did add 5 covariates specific to ocean species: bathymetry, bathymetric slope, monthly chlorophyll, monthly sea surface temperature, and monthly sea surface temperature anomaly. So we should be getting a lot more ecological signal, but we just don't have the data volumes over open ocean as we do on land. So it's going to be a data integration challenge for us and everyone in the future to model these species well. The other question I got was related to predictive performance metrics and quality. So I just wanted to go over what's in a little bit more detail, what is available in the R package. So in the current 2022 version, they on the right, I've listed them, and they're kind of broken down by the different types of predictions we're making. So if we're predicting presence absence, encounter rate, relative abundance, and the count estimate, we have these different metrics that are available. And on the left, you can see how we make these available. This is a map of that, but we make it available as a raster TIFF at 27 kilometers. Showing the spatial patterning of a particular performance metric. This one is the Bernoulli deviance, which in my opinion is one of the best for evaluating the encounter rate or occurrence rate. It has a fairly easy interpretability, as in if the value is over 0, your model is performing sufficiently. But you can see that there is a variation in pattern here. And you could use that information potentially to guide your decisions about using the data in different parts of the world. For resident species that we're looking at here, we provide one raster layer for the whole year, and that's based on the methodology that we model residents. They use all the data from a full year. For migrants, however, and this isn't the best example because it's a globally distributed species, but we do provide this information on a weekly basis. And so you can see that here you could access that information weekly if you want to. We did also revamp our predictive performance metrics that we are providing in this upcoming version. And I'm just highlighting a few of them here because we've added a bunch, but a number of ones that are a little bit easier to use. One is a normalized PR AUC, precision recall AUC. A lot of people are familiar with that metric in machine learning. And then if you take it and compare it to prevalence, it's another fairly interpretable check of whether your model is performing sufficiently enough to trust your results. And that's just a 0-1 that gets smoothed out to a value between 0 and 1 in these products. And then similarly, both of these Bernoulli deviance and Poisson deviance values, if they're above 0, your model is performing fairly sufficiently. You can see here in this golden eagle example that there actually are places where that value drops below 0, and maybe you want to mask those results out. So it's there's still a lot to be done in terms of interpretability on these, and we're going to work in the next year to provide guidance on that. But for people who are familiar with SDMs and species distribution modeling and machine learning, they do provide a starting point. All right, back to you, Matt.
All right. I'm wondering if there are a few questions in the Q&A box. I don't know if any of those are worth answering now. For example, maybe you can answer this one. Why do the evaluation metrics change for the same location over time? It's this human behavior changing seasonally.
Yeah, so basically the evaluation metrics are going to change based on what the data is seasonally. So our modeling workflow breaks up the models for migrants into monthly chunks of time. And so if we're fitting models, with the month of May versus the month of February, what input data is in there is going to change. And so it depends on, whether people visited certain sites, who visited them, what habitats got sampled. All of that is going to directly influence that variability by month.
And maybe somewhat related to that, there's a question about sort of how does spikes in effort due to things like birding festivals affect the results?
Okay, yeah. So the one thing that we do in our modeling workflow that helps control for that is to grid sample the data. So when we're choosing the data that each model runs, instead of just taking everything, we put a 3 kilometer by 3 kilometer grid over the data and randomly sample one detection of the species and one non-detection. And so if there's a pulse of information coming in at a certain time, it's going to randomly sample that down so that it's not as strongly biased by that event. However, if you do imagine that like there's a place where nobody goes and then suddenly a festival comes in, that is going to kind of not necessarily bias it, but that, you know, I would say that what happens after, before and after the festival maybe is not as representative as the high coverage during a festival. But so it is somewhat controlled, but severe examples where you go from no data to a ton of data are still going to probably carry a little bit of bias.
All right, then maybe the one more before we continue on. Someone's asking about including nocturnal flight call checklists. Like, is there a chance those could ever be included in the model since they're not complete?
Yeah, I would say there's still some debate about NFC checklists. Certainly, if you're monitoring with a microphone, no. But if you are an individual person standing outside and you can hear those species and you report it as a complete checklist, then yes, we can. I think that's still a valid detection process that is as similar to everything else that happens in eBird. But putting out a monitoring microphone and doing all of your identification through some sort of software is just too different than everything else right now. We're have a long eye on integrating sound based identifications in the future, but we're not there yet.
Cool. Okay, well, I guess let's get into the R component. Essentially, I'm going to start by going over a whole variety of the other data products that are available beyond relative abundance, because last time we mostly just talked about relative abundance. And then I'll get into two applications. And maybe I'll just quickly show you what those applications are so you can kind of have a sense for what's coming up. So very briefly, the first one we'll do is a continuation of the migration chronology example that we gave last time, but we're going to extend it to be for multiple species, you know, comparing chronologies across species. And then also you can see here, there's sort of like error bars around the lines. So these are showing, you know, for each week of the year, what's the proportion of population within one of the US states for a set of six bird species. And then the other one is another common thing that came up in the Google form. People are interested in how to sort of identify like important bird areas or prioritize sites for certain groups of birds. So we'll show you how you can make a few different ways of making maps that highlight important areas for a group of birds. So in this case, this is, you know, areas of importance for grassland birds in Montana. Okay, now I will share my RStudio. So you should be seeing my RStudio window. I sent out an email yesterday with the R script that I'm going to work through today. A lot of people requested that I send that out in advance. And maybe I'll just ask Tom or Andrew to post that link. Okay, cool. In case you didn't get the email. And if you did, I did make a few minor tweaks this morning, so you may want to get that R script again, just in case. I do want to just highlight that, like, the intention, the reason I didn't send out the R script in advance is that the intention here isn't really to sort of code along with me at the same time. When we do smaller workshops in person, that's typically what we do because we can be there looking over your shoulder and sort of troubleshooting things as we go along. But with, you know, as many people as we have today, that's, you know, that's just not possible. So you're certainly welcome to code along with me, but just be aware that there's a risk that you're going to run into some, you know, some bug or some issue that's hard to troubleshoot. And there were several people that said last time that they spent so much time trying to fix some syntax error in their code that they just kind of like lost track of what I was talking about. So I would say unless you're very proficient at R and feel comfortable troubleshooting things, I would probably just listen to me, and then you can always go back and watch the YouTube video, and you know, you'll have the script to go along with the YouTube video after the fact, and you can pause the video as needed. So that's my recommendation, but obviously you're free to do whatever you feel most comfortable with. Okay, so this is just a continuation of the same R script that I posted before. So I'm going to load some packages. It's the same set of packages as last time. It's mostly the eBird ST package, and then some packages for dealing with like spatial data and stuff like that. Then I'm just going to skip down to part 2. We don't need to rerun all the stuff from part 1. Oops, sorry, I'm actually in the wrong script. I don't want to use the complete script. I want to use this blank one. Okay. So first, last time we used this eBird ST download status function to download the data products. This time I want to show you a whole bunch of other stuff that's available beyond just the relative abundance. So let's just use Golden Eagle again as an example. That's what we used for a bunch of examples last time. Now you may remember there's all these download underscore arguments that control what specifically gets downloaded, but there's one called download all, which will just get everything. And also remember there's this dry run argument that will not, instead of downloading everything, it will just print a list of all the stuff that's available. So let's run that. Okay, so you'll see there's this big long list of all the stuff that's available for Golden Eagle. And it has a directory structure that's kind of meant to be intuitive. So you see it starts with the prediction year 2022, then the 6-letter species code. And then there's directories for each of the different broad categories of products. So at the bottom here, we have all the weekly data products. And they're all geotiffs. So that's raster data. So, estimates over a regular grid in space. And these will all have 52 layers, one for each week of the year. If you look, there's essentially 3 files for every data product related to and they'll say 3 kilometers, 9 kilometers, or 27 kilometers. So for each data product, we provide these 3 different resolutions. The 3 kilometer one is sort of the native resolution. That's what you should use in most cases, but in some cases, it may just be too computationally challenging to work with that data. So that's why we provide the lower resolution data. Okay, then you'll see there's a whole bunch of different data products. So this one here that says abundance underscore median, that's what we worked with last time. That's the same stuff that's on the eBird Status and Trends website. It's the median relative abundance on a weekly basis. But then you'll see above and below that, we also have a lower and an upper version of that. Those are the confidence limits on that relative abundance estimate. So it's the 10th and 90th quantiles of relative abundance. Below that, you'll see we have here occurrence and count estimates. You'll recall from Tom's presentation, or Tom's webinar a while back, that the models we use to estimate relative abundance are like 2-stage models. We estimate occurrence, and then we estimate count conditional on occurrence. And we actually, although you can't see those occurrence and count estimates on the website, they are available in the R package. It's sort of a more niche product. Not a lot of people use that, but it is there if you want it. And then finally, we have at the bottom here, a proportion population. So this is just a transformation of the relative abundance, where we take the relative abundance raster, we calculate the sum of all the cells across the entire, and we divide the abundance raster each cell by that number. So every cell, you get a value corresponding to the proportion of the population. So it'll typically be a very small number because there's many cells. That's a useful data product to use if you want to calculate like proportions of population within regions, because you can just sum cells that fall in within regions. We go up to seasonal data products, which are all these ones here. Oops. Oh, actually, I was going to load a few things. So I'll just say, if you want to actually, download all these data products, which I don't really recommend you do, take a long time. And after this webinar, you would just run this without the dry run. Let's load that portion of population, 7-kilometer resolution. Which one we used last time? We'll say golden eagle. Oops. For population, the way we specify that is with the product proportion population. And you can see you're doing
Matt.
Can you still hear me?
Yeah, you started breaking up. We were trying to decide when to interject, but your audio and video got really broken up there for a while.
Oh no. Okay. Give me 1 second.
The Q&A just hit for us. I don't know what, there was some sort of lag. So just give us all a minute to pause here and we'll maybe answer a question live. Well, Matt, do you feel like you're ready?
Yeah, can you hear me okay?
Yeah, your video is still laggy, and on Zoom it's showing red connection for you.
Okay. Try.
I'll just take a live question here while Matt works on that. Someone asked, can you describe how the 9-kilometer and 27-kilometer grids are aggregated from the 3 kilometers? So they are, wow, I need to remember how we do that. It's pretty straightforward if I remember. It's just a mean aggregation of if we take 3 cells on a side to aggregate up and just calculate a mean value. Okay, Matt, your connection just switched to 5 white bars, so maybe it's better.
Okay, we'll hope for the best here. All right, so I'm going to try sharing again.
You may also, it may make sense to just have you turn off your video and just do your screen share and audio too, if it becomes a problem again.
Okay, yeah, I just turned off my video. All right. So let's just go back. I talked about this line here is how you download all the data products for a species, which in general I don't recommend doing because you probably don't need them all and it's a lot of data. And then we were talking about how to load. I talked about all the weekly products, and I wanted to just show you how you would load the weekly proportional population product as just one example. Okay, so I was saying you would say product equals proportion of population, and then resolution equals 27 kilometers, because in this case, we want to load the 27-kilometer data. And let's just call this proportion population. Okay. So now remember I said that each cell gives you the proportion of population within that cell. So in theory, if you sum across all the cells, it should sum to 1. And we can do that using the global function, which is a Terra function that applies some operation to all the cells. So we'll give it that proportion of population raster. We'll say function equals sum, and we'll remove the NA values. Let's see what happens. Okay, so this is a weekly data product. So there's 52 weeks here, and you can see that the sum for every week is 1, which is what you'd expect. This species is occurring in, at least in, if you sum across all the cells, you get the full population back. Okay, let's go back to showing the list of all the data products. And I want to talk about the seasonal data products next. So you'll see here for the seasonal data products, you have the same set of 3 different resolutions, 3, 9, and 27 kilometer. And then you also have the same set of products. So you have abundance, count, occurrence, and proportion of population. And these are basically just, you take the weekly data and you're summarizing across the weeks that fall within each season. So breeding, non-breeding, and then the 2 migration seasons. On the website, when you're looking at the seasonal maps on the website, you get the mean relative abundance across the weeks. And we have those here. For example, you look, golden eagle, seasonal abundance, mean. But some people also like to see the maximum value across the season. So we also provide that too. So you can either get the mean across the seasons or the max across the season. And finally, another thing that we provide that's not on the website at all is, in some cases, you may want to know something about just sort of ignoring the seasons entirely, just sort of what's the mean or maximum relative abundance across the entire year. So this can be really useful for, if you're doing some sort of like identifying priority areas. You might have a place that's like an important stopover for shorebirds, and maybe the birds are only there for like 2 weeks of the year, but it's, there's some huge amount of the population at that site for a couple weeks of the year. So these full year data products can be useful for that type of application. And I'll just show you how you would load those into R. So we'll call it abundance, full year, max. Same load raster function. We'll do golden eagle again. So we want to load the abundance, the relative abundance. And period, instead of seasonal or weekly, I'm going to say full year to get the full year estimates. And then let's get the maximum relative abundance across the full year. So if you look at this, it's a raster that has a single layer, and every cell gives the maximum value across all the weeks. So, each different cell will have a value from a different week. All right, that's all I want to say about the seasonal data products. And let's talk about some of the other ones. So next, there's the regional stats. This is the same thing that Andrew talked about, which you can get through the website. You can also load it in the R package. It's just called load regional stats is the name of the function, and you give the name of the species. Oops. It gives you the same data set. So for each season, for each region, whether it's a country or a state, you get that same set of summary statistics. So Andrew already talked about that, so I won't go into that one in detail. All right, I'm going to again show that list of data products. Let's scroll up even further. You'll see the next section is ranges. So these are range polygons, which are they're derived from the relative abundance. They're polygons that identify the the boundary between zero and non-zero abundance. Sometimes those could be useful if you want to just like make a map. And they're available both as sort of a raw version that has like, you can see the jagged edges from the cells, and then one that just has a bit of a smooth applied. So if you wanted to load these range polygons, the function is load ranges. You give the species name. Say, let's do the 27-kilometer ranges. And by default, it will load the smooth ranges. And this is just, this is a spatial polygons, or it's like an SF object that stores the polygons, and then you also get some associated attribute information. So these are the polygons for the breeding, non-breeding, and then the 2 migratory seasons. Okay, and finally, if you go even higher in the list, you'll see a section for PPMs. These are the predictive performance metrics that Tom spoke about. And finally, you'll see a section for PIs. These are predictor importance rasters. They give you some spatially explicit information about the environmental, about sort of the associations between either occurrence and count, and then the environmental predictors that we use in the models. So they can tell you something about the habitats that species are associated with, although they don't tell you anything about the direction. So they might say, for example, that a species is heavily associated with deciduous forest or something like that, but there's no way to tell if it's like more deciduous forest means there's a higher probability of that species occurring, or if more deciduous forest results in a lower probability of that species occurring. Okay, that's all I want to talk about going through these data products. Most people will be interested in just like relative abundance or proportion of population, so I want to mostly focus on that today. Okay. Unless there's any major questions we should stop for, I'll get into the first application.
There's one that might be worth live answering here just real quick. How do you interpret the proportion of population using the product? Higher numbers or higher proportion of the modeled population or the global population? I can answer that one. It's the modeled population. And it kind of takes some species biology to know whether we've modeled the whole global population. With golden eagle in Matt's example, no, that's the modeled population. But in the lower 48 of the US, that's the whole population.
Thanks. All right, the first example is that multi-species migration chronology example I talked about. So the goal here is the final goal is going to be to plot migration chronologies with uncertainty estimates for this set of 6 grassland species in the state of Montana. The set of species is sort of chosen arbitrarily. It's not super important, just for illustrative purposes, but these are species that occur in those types of habitats in Montana. So let's load that list. And since we're going to be working with Montana here, I want to load a boundary of the state of Montana. This is again coming from the Natural Earth R package, which we used last time. All right, so eventually the goal is going to be to loop over all the species and then calculate the chronology for each species in turn. But let's sort of build this up in steps. So let's start by just doing a migration chronology for a single species. So I'll say species equals, let's look at the final one, Western meadowlark. I'm storing the name of the species in a variable, and we'll see why that becomes useful later. First step is we need to download the data. So eBird download status. I'm going to give it the variable storing the species name. And then I'm going to use that pattern argument again to only download the specific file that I want rather than getting everything. In this case, what I want is the median relative abundance. And for this example, I'm going to use the 27-kilometer data products because we're dealing with a lot of species and the weekly data. It'll just make the example go a lot faster. Okay. In this case, I've obviously already pre-downloaded the data, so it doesn't have to download it. But next step simply is to load that median relative abundance into our R session. So again, use the load raster function. I'll give it the variable storing the species name. And then by default, it will load the weekly relative abundance. So I don't need to specify that explicitly, but I will say resolution equals 27 kilometers. And I'm going to call this median abundance, abundance underscore median. So that's now in our R session. Now the goal is going to be to calculate proportions of population within the state of Montana. We're using proportion of population, remember, because we're comparing between species. So we can't use relative abundance because of the detectability effect. So first, I'm going to calculate the total relative abundance across the whole range of the species. And I can use the global function to do that. I'll give it the median relative abundance raster. And I'm going to use function equals sum, and I will remove the NAs. See what that looks like. Right, so you see it's a data frame with a single row where each value is a total relative abundance across the whole range for that week. In this case, actually, I just want this as a vector, not a data frame. So one way to do that is just extract that one column that we're interested in. So I'll rerun this, extracting the column. And let's convert that relative abundance raster now to a proportion of population raster. So I'll call this POP for proportion of population. It's based on the median relative abundance. And it's super easy. You just take the median relative abundance raster and then divide it by that total. And in this case, Terra, the package that you use to deal with raster data, Terra understands that what this means is divide every cell of the median relative abundance raster by this constant value. All right, let's run that. All right, so now we have 52 layers. Each one gives you the proportion of population within the cell. The next step is we just want to extract all the values within Montana and then sum them up. And we can use that same function we used last time, which is extract, to do this. The first argument is that median proportion of population. The second argument is the polygon that we want to do the summarization over. We'll give it a function to apply to all the cells that fall in the polygon. And I'm going to say ID equals false because by default, this extract function adds some unnecessary additional column. Let's run that. And let's see what it looks like. Okay, so what this is a one row data frame with 52 columns, one column for each week of the year. If you had given it a set of polygons, for example, one polygon for every state, you would have had 50 rows, one for every state. But in this case, we just had a single polygon for Montana, so you get just a single row. It turns out that for most things in R, it's usually useful to have this kind of data in a transposed format, so where each row corresponds to a week. So I'm just going to do that transformation now. So I'll call the final data frame chronology. because I know we're going to add a bunch of species to this, I'll actually store the species name as a column. Now I want to have the week, a column for the week of the year. I can get that from the row names of the data frame returned by the extract function. So I can just say names, pop, median, region. And this is actually will give me just a character representation of the date. I actually want the date itself, like a date representation. So I can just use the as date function. It will do that conversion for me. And then I'll grab the median relative abundance. One way to convert a one row data frame into a vector is you can just use the as numeric function. Okay, see what that looks like. Great. So now we have a data frame with 52 rows, one for each week of the year. And then this median column is giving the median proportion of population within Montana. Now remember, we also wanted to add in the uncertainty estimates to this. So I'm going to go back up and I'm just going to modify what I already have to add in those uncertainty estimates. The first step is we need to download those additional rasters. Now it turns out that this pattern argument is pretty flexible. You can actually give it a regular expression, which is a way of expressing different types of string patterns. So I can actually, the syntax I can use is separated by this vertical pipe. I can give it also lower or upper. So this will download anything that's abundance underscore median or abundance underscore upper or abundance underscore lower. Then I need to load each of those lower and upper rasters. So we'll call this one abundance underscore lower. I'll say load raster. The species, which this variable, remember, stores the species name. Oops. The way you load the lower or upper is using the metric equals lower function or argument. And then remember, I also want to do this with 27-kilometer resolution. So this will load the lower confidence limit of the relative abundance at 27-kilometer resolution. I'll just copy and paste that and do the exact same thing for the upper confidence limit. So metric equals upper in this case. Let's load both of these. All right. Now, in the same way that we calculated the median proportion of the population, I'm going to do exactly the same thing for the upper and lower. And I'll just copy and paste again. So we'll say pop lower equals, we take now the lower confidence limit of the relative abundance and then transform that to a proportion of population. And the same thing with upper. Okay, let's just run these two lines. And it's, again, just going to be copying and pasting. We already have the median proportion of the population in the region, and we want to now do the lower and upper confidence limits. So I'll say lower and upper, and then I need to switch to be referring to the lower and upper rasters accordingly. And I'll run all of this. So each of these looks, basically the same as the, there are also these one row data frames that give you for each week of the year, the median, lower, or upper relative abundance. And when I do this transformation, I'll do it such that the median relative abundance, sorry, the median proportion of population, and then the lower and upper confidence limits are as stored as columns. So I'll say lower equals as numeric POP lower region, and then upper equals as numeric POP upper region. Let's see what that looks like. So now we have a data frame, looks like this. Species name, the week of the year as rows, and then the median proportion of population as well as the confidence limits. And I already have some R code here to do the plotting of this data. So let's just run that R code. And you can see at this point, it's just one species, but we'll add the other species shortly. This is giving you across the x-axis, the week of the year, across the y-axis, the proportion of population in Montana. And then for Western meadowlark, you get this migration chronology showing sort of when the species shows up in Montana as it migrates north, and then when it leaves in the fall. The last step is just to wrap all of this in a loop, to do this exact same thing, but for each of the 6 species. So rather than this species equals meadowlark, we'll just change this to 4 species in, I think it was called grassland species. And then I'll wrap everything here in the for loop. I'll also select all of this and I'll just re-indent it. So now we're looping through the for loop and doing this for each species. The final thing to do is just, as it stands, we're overwriting that chronology data frame each iteration through the loop. Instead, we want to append to that data frame. So there's a whole variety of ways you could do this, but one way I often do it is I'll just start with just giving an empty object as chronology. And then, oops, down here, I'll use bind rows to append each species to this data frame. Okay. It'll take a couple of seconds just to go through. It's processing each of those 6 species in turn. Okay, we're finished. Let's look at the results. Great. So this now has, that's basically the same plot as we had for the Western meadowlark, but now it's for all 6 species. And you can see that some species like Western meadowlark, and here in yellow now, they arrive pretty early, sort of mid-March, and they leave pretty late in October. Whereas other species like upland sandpiper, which is in orange, it's in this region for a much shorter period of time. It arrives the beginning of May and leaves sometime kind of towards the end of July. One thing I want to highlight about this plot, we sort of intentionally picked a somewhat problematic species. So you see here, there's this Sprague's pipit in purple. It has this massive spike at the end of August. And I think Tom mentioned earlier, so sometimes you have to kind of use your intuition and your knowledge about the ecology of a species when you're looking at results to make sure they make sense. This seems a little odd. And one thing about Sprague's pipit is during migration, it can be kind of hard to detect. So that sort of gives me a little bit of suspicion something weird is going on. And I just want to quickly jump over to where is it? I want to jump over to the Status and Trends website for this species. let's go to look at the weekly results kind of in September and step through this animation. Oops. Okay, so you can see there's a big blob in Montana and parts of Canada during the breeding season. And then as we go through and they start to migrate, you see there's sort of times when they kind of disappear a little bit. Basically what's happening is, like right around here, they are migrating through a broad swath of the US, but they're really hard to detect. So we're actually kind of missing a big chunk of the population. But in Montana, people are still picking them up. So this is why that spike is not real. It's basically an issue with detectability. Okay. All right, so before I jump to the second application, are there any questions that we should address?
Yeah, we're good right now.
Okay, I'm just gonna take a sip of water to give everyone a chance to kind of get caught up. Has my video been okay? Haven't had any other issues?
Your audio has been fine ever since you turned video off, so probably
Okay.
best doing that.
Cool. All right. So the other application that a few people requested in the Google form was stuff in this kind of broad category of like site prioritization or identifying areas of importance for a species. This is a really common application for status and trends data products, and it's I think an area where status and trends can really shine, because especially for areas like the US, we have a huge number of species, and we're showing their whole range. So for this example, I'm going to use that same set of six grassland species in Montana, and I'll show you a couple different ways that you can do this, to basically make a map of important areas for this set of six species. The sort of first and simplest way you could do this would be just to literally make a richness map. A map where every cell is the number of species that you would expect to see in that cell. And so first I'll do that. In this case, rather than building the loop up from scratch, I sort of did that for illustrative purposes in the first example. I already have the template for the loop here, so I'm just gonna go right into it. So again, basically the goal here is going to be to loop through the species and create a map that, rather than having something like relative abundance, it's just binary. It just has a 0 or a 1 depending on whether the species is present or absent, and then I'll just add up across all the species. So first step, I got to download the data. So eBird SC download status. I'm going to use the species, the variable that we're going to loop through over the species names. I'll say pattern. Again, this is just downloading the data that I actually need rather than downloading everything. In this case, what I want to do this for the breeding season. I didn't mention that earlier. So I'm going to download the seasonal mean relative abundance at 3 kilometers this time. Then I want to load the seasonal relative abundance raster and subset it to just the breeding season. So I'll say abundance equals load raster species. I'm going to say, in this case, I want to use the seasonal period. And I don't need to specify relative abundance because that's the default. And I can use this subset function from Terra to just pull out the breeding season. Okay. And then next, right now we have this is for the entire globe essentially. But what I'm just interested in Montana. So I'm going to crop the raster to a boundary around Montana, and then I'll use the mask function to remove any cells that fall outside of Montana. So I'll call this abundance masked. So I'm going to mask and crop at the same time. I want the raster that I want to apply that to is called ABD for abundance, and then the polygon that I want to use for the cropping is that Montana boundary, and then I need to also give that polygon to the to both the crop function and the mask function. And finally, this is a relative abundance raster. I want to convert it to just a binary presence absence raster. You can just do that by converting everything above 0 to a 1. And I'll store that in this variable range Montana, which I'm using to iterate, to collect the result as I iterate over them. So I'll say range Montana. This is a list, and I'll store the results as an element of the list, named according to the species. So all I do is abundance underscore mask greater than 0. And the Terra R package knows when you use this syntax to apply the operation greater than 0 to every cell of the raster. Let's run this. Again, we get a bunch of these sort of messages just saying that I've already pre-downloaded the data. So what this looks like, it's a list with 6 elements. Each element corresponds to a species. So in this case, Western meadowlark. And it's binary. So for the breeding season, it's giving you a true or a false, whether the species is present or absent, or predicted to be present or absent. To calculate the richness, I just need to do a cell-wise sum over these rasters, over these 6 rasters. I'll just show you that I can take this from a list of 6 rasters to a raster with 6 layers by just using the rast function. So rast range underscore Montana. This is now a raster with 6 layers. And then I can apply the sum function. This will do a cell-wise sum. So for every cell of the raster, it'll sum across 6 species to give you a richness value for that cell. I'll call it richness. And I'm just going to use the default plotting function from Terra to show you roughly what this looks like. You can say plot richness. It's not going to be a nice map, but it'll at least let us look at it. So this is showing you at 3-kilometer resolution for the state of Montana, between, how many species are in each cell, between 0 and a total of 6. Here are the yellow values. In the northeast are areas that have all 6 species. So that's kind of like the roughest approach to doing this. Just gives you, it's using the presence and absence of species, but it's not really using like the full power of status and trends, because we have information not just about presence and absence, but also relative abundance. So, there may be some cells in this raster that have, a very low like they have, say, Western meadowlark, but at a very low abundance, and other cells that have a huge number of Western meadowlarks. And typically in a prioritization exercise, you would want to prioritize those high abundant cells more than lower abundant cells. So one of the sort of simplest ways that we can do that is do something very similar to what I just did, but instead of summarizing the presence absence rasters, let's use the proportion of population layers. We use proportion of population again because we're dealing with multiple species at the same time. So we can't compare relative abundance, but we're safe to compare proportion of population. So I'm going to essentially do the exact same thing, but with proportion of population. So I'll do, eBird ST, download, status. I'm going to give it the species name. And in this case, I'm going to use pattern equals proportion population seasonal mean 3 kilometers. So just download that specific proportion of population seasonal raster. Let's load that raster into R. I'll call it proportion population. I'll say load raster. And I'm going to give it the species name. Product now is proportion population period equals seasonal. So that will do the loading of the raster stack that has 4 layers, one for each of the 4 seasons. But really all I care about is the breeding season. So I'll subset to the breeding season. And the final step is just to, again, crop and mask that to the state of Montana. And then store it in this variable I'm using to keep track of all 6 species. Proportion, population, Montana. Oops, I need to store it as an element of the list. I'm going to do mask and crop at the same time again. Proportion population, that's the raster layer that I want to apply this cropping and masking to. Montana boundary is the polygon I want to use to do the cropping, and I have to provide it once for the crop and once for the mask. All right, let's run that for all 6 species. And in this case, you could take a sum or a mean. I'll do a mean across a mean proportion of population across the 6 species. I'm going to just call this importance because it sort of gives you, it's a raster that's, it's a metric that's giving you the something about the importance of each cell to that group of 6 species. So I'll say mean. Remember, I have to convert the list to a raster stack. Proportion population Montana. And as always, just removing the NAs. Okay. Let's take a quick look at what that looks like. So I'll just plot it. All right, I'm going to make this a little bigger. So this is giving you for each cell the mean proportion of population across this group of 6 species. It's a number that's kind of hard to interpret. It's not really like a value in an absolute sense, but it gives you a sense for ordering, like the relative importance of different sites. There's also a lot of, out here in the western part of the state, there's a lot of zeros here. So let's actually just remove the zeros. So I'm going to use the same variable name. There's a function called if-else in the Terra package. It's similar to the if-else function in base R. And I can say basically, if the importance, if the cell value of this importance raster is equal to 0, just convert it to NA. If not, leave it as is. So now let's see what that looks like. Okay, so it's basically the same thing, but I've just removed the zeros. Now at this point, you could use this as is. A lot of people like to highlight just the most important areas. So one option in this case is to sort of pick some cutoff and then remove everything below that cutoff. So for the sake of the example, let's define the cutoff as the median value. So we'll remove the bottom 50% of values. So I can calculate the cutoff by using the global function with the quantile function, this gives us the 50th percentile, which is the median. And I'll show you what that looks like. It's just some kind of like arbitrary value, which is the midpoint of all the cells in this raster. Again, it's stored as a data frame. What I actually want is just a value, like so I'm going to use the as numeric function to just extract the value itself. And then I can just use that same if-L function, but now I'm going to say if importance is greater than the cutoff, leave it as is, but if it's less than that cutoff, convert it to NA. Okay. And lastly, just to make a slightly nicer map, I have some fairly complicated code here that will make a bit of a nicer version of the map. So this is what it looks like. This has removed that, the bottom 50% of cells that have low importance. And we're left with just this more important 50% of cells. And the most important cells here are in yellow. And the legend here, because the actual absolute values aren't all that meaningful, I've just labeled it as low, medium, and high relative importance. That's the end of the example. I will say that there's a whole bunch of different ways you could do this. For example, you could use a 70th percentile cutoff if you want to identify, even higher importance areas. You could also use, for example, rather than the breeding season, you could use like the full year maximum relative abundance or something like that. But this gives you sort of like the most basic way you could do this.
Matt,
Okay.
was just a kind of an elaboration on that. Caitlin was asking why to use the mean of percent of population instead of the sum for the cross species aggregation, and Andrew and I were discussing that both are certainly plausible. Would give you
basically
similar...
the number in an absolute sense means nothing anyway, so you could do the mean or the sum. It's just a scaling factor essentially. We're close to that 90-minute mark, so I want to leave some time for questions. Before we do, I'll say I have a couple of exercises here, which if you have the time, it'd be awesome if you do them, especially because if you do them and you have a problem, you can send me an email. Maybe Andrew or Tom can just post my email in the chat. And I can try to help you with them between now and next week. They're basically just redoing the two examples I just did, but with slight modifications. And the last thing I'll say is that next session, we're going to move on to the trends data products. And I have another Google form, which Tom or Andrew can post the link for, which asks for both questions that you may have had from this session, but also for examples on the trends side that you're interested in. Some people already asked for some sort of trends related questions in the previous Google form, but we're going to leave those to next time. So I think that's all I have to say. And I guess we can do any other questions about anything that we've covered today.
Okay, Matt, the form's up. Got one question here, Matt, specifically. Can you elaborate a bit on the use of mass, crop, and extract functions for that kind of thing?
So, okay, the first thing I'll say is that the difference between mask and crop. So all of the rasters that we release are always for a global extent, just for consistency. So like even if, you imagine like some super range restricted species that occurs in like one tiny little location in the Andes, maybe it only has like 5 pixels that have non-zero values, but we're always going to give you the full raster. But anytime you want to, make a map or do an analysis, often, you don't actually need the full global extent. So it can be useful to just crop it, just remove everything outside of some certain region. So that's what crop does. But crop just crops to a box, like a rectangular box. So Montana is not a perfect rectangle. So if you crop to Montana, it will crop to a box that surrounds Montana, but you're still going to have values that are just outside the edge of Montana, but still within that rectangular bounding box. So the mask function then removes those additional values. It converts them to NAs. It's, basically the crop function is mostly just for like making maps and stuff like that. Whereas the mask function removes values, and that's key for the analysis. Extract it also takes a polygon, and it will find all the values of the raster that fall within that polygon and do some operation. So like a sum or a mean. Now, everything we did, like when you're calculating proportions of populations, you can use the extract function, or you could mask the raster to remove the values outside of the polygon and then just sum them up. It's sort of, you can choose which one you want to do. The benefit of extract is that extract is smart enough to know about cells that are only partially covered by a polygon. So if the polygon boundary goes to the middle of a cell and you use sum, it will only sum up half the value of that cell rather than the full value, whereas crop doesn't know about that.
Matt, Andrew and I have been busy answering questions, so we haven't been following. For the Montana example, were those the global proportion of populations or the Montana proportion of populations?
Those, okay, yeah, those were the global modeled proportional population. I talked about there's a whole variety of different ways you could do this. You could instead calculate a proportion of population relative to a different region. So the example I think I have as an exercise is to do that same thing, but instead of calculating the global proportion of the population within Montana, calculate the US proportion of the population within Montana. So in that case, you first mask everything to the US and then convert the masked relative abundance to a proportion of population. And it really just depends on your particular application.
So there's a question about understanding changes over time. So this is a good tee up for trends, although we can't answer this specifically. Somebody wants to know if you understand the changes in phonology of the application one, the migration chronology. Could you download previous versions and do this? No, I'm going to say is the most reliable answer here, because a lot of the changes of the status versions that we produce year to year are primarily due to tweaks in methodology, the features that we include each year, and the prediction values that we've tweaked. So that's really more for a trends product, which we'll talk about next week. However, our trends products right now are primarily for stationary breeding or non-breeding periods, so they wouldn't cover a phonology. That is something at this point that you would have to analyze raw eBird data to generate that sort of analysis. There was just a good paper out of sorry.
I'm jotting down a paper from Nature Ecology and Evolution
Thank you.
that does this from 2021. So I'll share that over the question and answer.
I did see someone posted, Connor looks like posted a function that he wrote that will download multiple species at the same time. As opposed to having to loop, it's just one function that will download a bunch of species. This is maybe something we could just add, I guess, to the R package. I guess the reason I didn't do it is I'm trying. I suppose I want to discourage people from just like, running the function for like every species and just downloading like 12 terabytes worth of data. By making the function download one species at a time, the idea was to maybe be a little more intentional. It's also useful for us that way because it's useful for us. We can see which species and which data products are getting downloaded more. So sometimes it's useful to us to be able to understand specifically which species are being downloaded more. But maybe after this, we could talk about the potential of just making that functionality available directly in the R package.
Answering question live here, and I think Andrew maybe will take this one. Are you aware of people using the chronologies or proportion of population estimates in bird groups, such as species at risk in conservation context? What would be your concerns in doing that?
Yeah, so this is something that we see quite a lot of, and it's a good thing. This is an appropriate use of these sorts of data sets. One thing that I really like about the seasonal proportion of population estimates and migration chronologies is that they let you see how your responsibility or your stewardship responsibility within a region changes across the full annual cycle of the bird. For example, we see some cases where, Michigan might not think about its responsibility with tundra swans or trumpeter swans. I forget which one it was, but something like 15% of the world's trumpeter swans or tundra swans migrated through Michigan during 1 week of the year. And so it gives you a view of not just where, but also when your area might be especially important for species. And we see this with a lot of applications, especially within the US in like state level, state wildlife action plan planning and management prioritizations.
Thanks for that, Andrew. That's exact. There was a number of people who commented in the previous Google form that we could provide more like motivation for examples, which I think I probably didn't do quite enough, but that's super useful.
There were a number of questions here in the Q&A about the spatial data that you can access to analyze, like the examples that Matt gave with Montana. There were people who were interested in city boundaries, county boundaries, the eBird regions that are available. So I would say that after natural earth data, your next best resource is called GADM, and I'm going to put a link to that in the chat. GADM is what eBird did source their regions from once upon a time. We're not up to date with the most recent version of GADM, but GADM is a open source effort to describe as many administrative regions as possible. And you can go download smaller chunks of the data there. I feel like I've tried to use a GADM R package before and I've hated it. So I would suggest just downloading the spatial data directly.
Yeah, I think Tiger is also another possibility.
Yep, you could use Tiger. So there's a lot of those resources. eBird specific regions are not available for download. GADM is going to be your best analogy for that.
But also just like even more generally, like if you have a shape file, you can just load that into R. There's a function called read_sf. And that will just, for whatever custom region you want, you can load that into R and then basically just do exactly the same thing.
Oh shoot, I didn't mean to dismiss that one. Hold on, we got one here. Is it possible to analyze the phenology of all bird species in a single year? I'm sorry, a single area for 1 year. So I think very similar to what you've done, but phenology of all species. I don't see why not. It's just kind of a computational limit.
Yeah. I mean, for any species that we have the data for, you can do it. That does remind me. There was a number of questions about last time about kind of like being able to just identify for a given polygon, for example, or a given region, like what species are there according to status and trends. which,
Right.
to be honest, like we don't have a great way of doing that at the moment. Because essentially, you would have to like download the data for all the species and then sort of go through each one to identify if it's there. It's something that in the future we might be able to add that functionality, because I know it is really important to people. I will say like the maybe the closest thing is just on the eBird website. If you go to the Explore tab, you pick a region, for example, like Belize, and you can click on. There's a variety of ways to get this, but illustrated checklist is one option. It'll give you a full list of all the species available. The one catch with this is that it will include things like, like this Canada goose. The species doesn't really occur in Belize. You can see there's just a couple of little dashed lines here, which, maybe it showed up once or twice as a vagrant. So you'll end up with a bunch of vagrants as well. This is just
You can download the bar chart for this, and then kind of calculate like a maximum value and threshold it to drop those. I've done that for defining which species from which regions we want to run. So it's something.
So those are available here, also here on the left bar chart. And it's sort of the same data, but it'll allow you to download it. And then, like Tom's saying, you could subset it to try to remove those vagrants. But hopefully, at some point, we'll have some status and trendsy way of doing it.
Actually, Matt, do you mind just popping up your screen and doing the draw polygon tool on the S&T page? Somebody had a question about, is it complicated to make a custom polygon? The general answer is yes, like making polygons from scratch in like software like QGIS or ArcGIS requires, a certain level of GIS skill, but we have added an ability to draw fairly simply shapes for the seasonal and weekly animations. Matt's going to show here how you
this little button here on the bottom left of the toolbar, and then you can pick some arbitrary area, and then if you scroll down here, it'll give you the proportion of population for each of the 4 seasons, or on the weekly tab, it'll give you that migration chronology. And you can draw multiple polygons too if you want. And then it will give you kind of like 2 different chronologies.
Someone is asking whether there's a limit to the amount of data you can download. No, there's no throttling on the status data package. Aside from your connection, it uses Amazon Web Services as a backend. So if you make 1 million calls really quickly, something might reject that, but otherwise you can download away.
Someone commented, Caitlin commented, you can also download the shape, I think. You can't download the shape, but you can download the values. So you can download the proportions of population within those. Or is that not true?
Yeah, no, you can download the shape. It comes as a CSV file in a well-known text definition, which is
okay.
kind of like simplest format, and you get
There
your
you go.
shape. So
Nice. That's great.
There's a question about analyzing phenology diversity. I'm guessing this is looking at the diversity in arrival and departure times for migratory birds.
Oh, yeah. I hadn't thought about that.
Yeah, and so something like this would either require doing that migration chronology that Matt shared with us, doing that across a big list of species. But another way to do that is to use the bar charts from the eBird website, not the eBird Status and Trends website, from the eBird website itself, just to start looking at when does the species tend to arrive and when do people start reporting it. That just is a summary of raw eBird data, but it might be able to give you some of the information that you need.
Yeah, otherwise, if you're going to analyze that sort of thing, you're going to want to do that richness step that Matt did with Montana by week.
and then calculate a chronology off of that product if you want to look at like species richness over time.
So
So it's doable, but yes, requires a little more effort.